In this module, Data Analysis, this module explains how safety data are analysed and, just as importantly, how far the results can be trusted. It covers the main data sources and their limits, the biases that shape spontaneous reporting, the 2x2 table, and the disproportionality measures built on it, and the epidemiological study designs used to test a signal properly. Learning outcomes. Compare the main sources of safety data and explain the strengths and limits of each. Explain the biases affecting spontaneous reporting, including underreporting, the Weber effect, and notoriety bias. Construct a 2x2 contingency table and calculate PRR and ROR from illustrative reporting data. Interpret IC and EBGM and explain why disproportionality thresholds are convention rather than regulation. Data sources for safety analysis. Spontaneous reports from schemes such as Yellow Card, Vigilance, FAIRS, and Vigibase capture suspected reactions in real-world use. Clinical trial data give clean exposure records and a comparator arm, but small numbers, short follow-up, and selected patients. Electronic health records hold rich clinical detail and a usable denominator, but coding varies and a prescription is not a dose taken. Claims data cover very large populations with reliable dispensing records, but they record billing categories rather than clinical judgment. Spontaneous reports, strengths and limits. They cover every patient, every indication, and the whole marketed life of a product, including groups excluded from trials. They are cheap and fast and remain the only practical way to detect very rare type B reactions after authorization. A valid case needs 4 things: an identifiable reporter, an identifiable patient, a suspect medicinal product, and a suspect adverse reaction. There is no denominator, so you cannot calculate incidence or a true rate from spontaneous data alone. Bias in spontaneous reporting. Underreporting. Hazel and Shakir's systematic review of 37 studies found a median underreporting rate of 94%. Weber effect. Reporting was described as peaking around the 2nd year after launch, then falling away as the product became familiar. Hoffman and colleagues analysed 62 drugs in FAIRS and found most did not follow that pattern, so treat it as a hypothesis. Notoriety bias. Pariente and colleagues showed that reporting of a drug event pair rises sharply after a safety alert. The 282 contingency table. Every disproportionality measure starts from the same 4 counts, built for one drug and one event at a time. Cell A holds reports that name both the drug of interest and the event of interest. Cell B holds reports naming the drug but a different event. Cell C holds reports naming the event with other drugs. Cell D holds reports naming neither the drug nor the event, which is normally most of the database. PRR and ROR, an illustrative worked example. Illustrative figures only, not real product data. Drug X has 1,000 reports in total, of which 30 name event Y, so all other drugs together have 99,000 reports, of which 1,485 name event Y. So A30, B970, C1485, D97515, PRR compares proportions. 31,000 is 3.0% against 1485 99,000, which is 0.5%, giving a PRR of 2.0. ROR compares odds, 3970 divided by 1489 which equals 29950 divided by 14050 or 203. IC and EBGM, Bayesian disproportionality. Both compare the observed number of reports with the number expected if drug and event were reported independently. Expected count here is drug total times event total divided by database total, 1,000 x 1515, 100,000 1515. The information component is the base 2 logarithm of observed over expected, log to 30 1515, log to 198, which is about 0.99. An IC of 1 means twice as many reports as expected. An IC of 0 means exactly as expected, and a negative IC means fewer. Thresholds are convention, not regulation. A commonly cited PRR convention is a PRR of at least 2, a chi-square of at least 4, and at least 3 cases. These are working conventions chosen to balance missed signals against false alarms. They are not legal requirements. The EMA's UDRA vigilance screening guidance uses the reporting odds ratio, with the lower bound of its 95% confidence interval above 1. It also applies a minimum case count of 3 or 5, depending on whether the product is under additional monitoring. The Signal Detection Workflow and EVDS. Signal management under GVP Module 9 moves through detection, validation, confirmation, analysis, and prioritization, assessment, recommendation, and communication. Statistical screening produces a long list of drug event pairs. That list is a starting point for human review, not a conclusion. EVDAS is the UDRA Vigilance Data Analysis System, which supplies predefined tables, graphs, and the electronic reaction monitoring report. Marketing authorization holders and regulators screen at defined intervals, with the frequency reflecting the product's monitoring status. Qualitative review and signal triage. Statistics tell you where to look. Reading the individual cases tells you whether there is anything worth pursuing. Assess case quality, time to onset, dose relationship, de-challenge and re-challenge information, and whether alternatives were excluded. Assess confounders, underlying disease, concomitant medicines, and whether the event is a known feature of the indication itself. Look for duplicates, literature cases counted twice, and clusters arising from a single reporter, program, or study. Study designs in pharmacoepidemiology. Cohort studies follow exposed and unexposed people forward in time and can estimate incidence, risk, and rates directly. Case control studies start from cases and suitable controls and look back at exposure, which makes them efficient for rare outcomes. Nested case control studies draw both cases and controls from inside a defined cohort, keeping efficiency and a known source population. Self-controlled case series compare risk periods with other periods in the same person, so each case acts as their own control. Confounding, channeling, and immortal time. Confounding occurs when a third factor is associated with both the exposure and the outcome, and distorts the apparent association. Confounding by indication is the central problem in drug safety. The condition being treated is itself linked to the outcome. Channeling bias is a specific form, described by Petrie and Urquhart, where newer medicines are directed to higher-risk patients. Immortal time bias arises when follow-up includes a period in which the outcome could not, by design, have occurred. Absolute versus relative risk. Relative risk divides the risk in the exposed group by the risk in the unexposed group. How many times more likely? The odds ratio divides the odds in each group and closely approximates relative risk when the outcome is rare. The hazard ratio compares the rate at which events occur over time and is what survival analysis normally reports. Illustrative example: 12 events in 4,000 treated patients, which is 0.3%, against 4 events in 4,000 comparator patients, 0.1%. Incidence and exposure denominators. Incidence proportion, or risk, is the number of new cases divided by the number of people at risk at the start. Incidence rate is the number of new cases divided by total person time at risk, which copes with people followed for different lengths of time. In drug safety, the denominator is often patient treatment years, the total time patients were actually taking the medicine. Illustrative example: 15 events among 2,000 patients treated for an average of 1.5 years gives 3,000 patient treatment years. Visualization, reproducibility, and privacy. Good safety graphics show the distribution, not just a summary, time to onset plots, dot plots, and treatment emergent event displays. Plot counts alongside rates, label the denominator on the figure, and avoid truncated axes that exaggerate small differences. Reproducibility means another analyst can rerun your query and get your numbers. Version the extract, script the analysis, keep the log. Alcoa Plus applies to safety data as much as to trial data, attributable, legible, contemporaneous, original, accurate, and complete. That completes this module. The key takeaways and suggested further reading are summarized on the slide. The full references for this module are listed on the slide for your further reading.